This video explains how to use the equation C1V1 equals C2V2. This equation is very useful when you dilute a solution. In this equation, C1 is the starting concentration. Typical units are molar, micrograms per milliliters, etc. C2 is the final concentration. Try to use the same unit as C1 to make the calculation easy. V1 and V2 are starting volume and final volume, respectively. Common units are liter, milliliter, microliter, etc. Also, using the same unit makes the calculation easier. Let's practice. You have 50 xTAE buffer stock solution. You want to make 1 liter of 1 xTAE buffer by diluting this stock solution with distilled water. How much 50 xTAE buffer do you need? How much distilled water do you need? The TAE buffer is commonly used for agarose gel electrophoresis of DNA, and this dilution of a very common preparation in molecular biology labs. The point is how to assign these numbers. In this case, 50x means 50 times concentrated stock solution. 1x is a working solution. Therefore, C1 is 50, and C2 is 1. The units are the same. V2 is 1 liter, but V1 is unknown. We have 3 numbers out of these 4, and V1 can be calculated as 0.02 liters, which is same as 20 milliliters. We need one more step to answer these questions. V2 is the final volume, which is V1 plus water. Now we know both V1 and V2, and we can calculate the amount of water to be added. It should be 0.98 liters. The answer is 0.02 liters and 0.98 liters. Let's practice using a different question. This time, you have a DNA solution of 100 micrograms per milliliter. You need to prepare 10 microliters of DNA solution at a concentration of 20 micrograms per milliliter. Explain the process of dilution. Similarly, we can assign numbers. C1 is 100 micrograms per milliliter, and C2 is 20 micrograms per milliliter. The final volume, V2, is 10 microliters. Thus, V1 is calculated to be 2 microliters. Finally, the amount of water is 10 minus 2, which is 8 microliters. Now dilution should be an easy experiment for you.